Welcome back to all. Today we are going to discuss a part of immunology, that is kinetics of the antibody response. Actually, humoral immunity is mediated by serum antibody, which are glycoproteins secreted by B cell. B cells are initially activated to secrete antibody after binding with specific immunogen to specific B cell receptor, which are membrane bound immunoglobulin molecule expressed by B cell. Once antigen bind to the B cell receptor, B cell receive signal to make antibody against that particular antigen. The main purpose for making antibody is to eliminate antigen from the host. Now coming to what is primary response. When an individual first exposure to a particular immunogen, that the immunoresponse which is generated against that immunogen is called primary response. Primary antibody response may be divided into 4 phases. First one is lag or latent phase. It is the immediate stage following antigenic stimulus, during which no antibody circulated in the serum is detectable. There is no antibody is detectable in the serum. The length of the period is generally extended from 1 to 2 weeks. 1 to 2 weeks required to make antibody after primary immunization. Next phase is lag or exponential phase. In this phase, there is steady rise in antibody concentration in the serum. Next phase is steady state. There is an equilibrium between antibody synthesis and its degradation. There is equal amount of antibody which is synthesized or degrade. The last phase is declining phase. Here the concentration of antibodies in the serum decline rapidly. Now we will discuss what is secondary response. Second exposure to the same immunogen result in secondary response. In simple word, when the same immunogen second time infect an individual, the immunoresponse which is generated is known as second response. This second exposure may occur after the response to the first immune event has lived or has totally subsided. Also, it is also known as memory response. B and T lymphocytes that participated in this response are termed as memory cell. The primary response is slow and short-lived with a long lag phase and low titer of antibodies are detected, and it is do not persist for long time. But the secondary response is prompt, rapid, powerful and prolonged with a short lag phase and a much higher level of antibody that lasts for long period. In primary response, the first class of antibody is IgM, then IgG or another antibody classes, but there is a marked change in the type and quality of antibody in secondary response. There is a shift of class response known as class switching, IgG appearing at the higher concentration in secondary response, and then IgM. Actually, IgM is not found in secondary response. Other antibodies found in secondary response are IgA and IgE. So IgG, IgE, IgA are collectively referred to as secondary class antibodies. Thank you.